So this is our first lecture where we are having an introduction to the concept of what? Hemoglobin synthesis. Also the structure of hemoglobin, then also talking about the functions of this hemoglobin in the body, right? Which is mainly transport of oxygen, then also transport of carbon dioxide. Okay. So looking at this is basically heme formation, heme synthesis. So hemoglobin is a protein containing iron that facilitate the transport of what? Oxygen in red blood cells. It is part of the red blood cell that is composed of a red pigment containing iron and non-iron components. So almost all vertebrates contain hemoglobin with the sole exception of what? A fish family known as what? Canictidae. Alright. So the name hemoglobin is derived from two words. So you're basically having what? Hem coming together with globin, forming hemoglobin. So the iron present in hem is confers to what? The red cell. It confers to the red cell a red pigmentation. So it's the iron that actually gives what? The red blood cells that they are characteristic red color. Alright. So alright. And this actually represents the heme derivatives, while the non-iron containing part is composed of proteins, particularly what? Polypeptides, and this actually represents what? The globin derivatives. Okay. Another picture showing the different types of what? Steps in heme production. So generally, each hemoglobin is a tetramer made up of two pairs of globin polypeptides linked to a heme moiety. So the two pairs of unlike polypeptide chains is made up of what? One chain of each pair of what? Alpha or alpha-like, and the other one of a non-alpha, which could be beta, lambda, or gamma. Okay. So certain residues in the amino sequences of each polypeptide chain appears to be critical to stability and function. So there are different hemoglobins that are synthesized at different stages of human development, right? So one thing is hemoglobins that are found at the embryonic stage are different from the ones that are found at the childhood stage, and are also different from the ones that are found at adult stage. So sometimes in some disease condition, if you are seeing a hemoglobin which is not supposed to be found at a particular stage, it helps you to confirm that that disease condition exists there. So in embryonic life, before the 10th week of intrauterine life, there are usually three types of hemoglobins which are produced. This is hemoglobin Goa 1, hemoglobin Goa 2, hemoglobin Portland. Then during fetal life, we have the hemoglobin F, which is alpha square and that one square. Then in a normal adult life, you can be having hemoglobin A or hemoglobin A2. Right? So hemoglobin is synthesized in a complex series of steps, but generally the steps are broken down into what? The synthesis of the heme, then synthesis of what? Globin, right? So the heme synthesis involves series of steps in mitochondria and also the cytosol, right, of immature red blood cells. Because the mature red blood cells, I think they lose the mitochondria or something, so heme cannot be formed. So we say that, however, globin synthesis takes place by the ribosomes in the cytosol. Right? So this is just introduction to the concept. We have much more videos coming on it, alright, so stick with us.